Okay, why don't we go ahead and get started? I want to welcome everyone to Nathan Lazar's PhD Symposium, which, as many of you know, is one of the steps along the way to doing a dissertation, which usually involves gathering background information, starting to formulate hypotheses, research plans, etc. So Nathan is a student in the BCB track. He did his undergraduate work at Ohio University, sorry. And then got a master's at Portland State and has been in our program, both in the PhD program and funded by the NLM training grant. So, take it away.
All right. Thank you all for coming. It's nice to see everybody here for me. So it's a symposium talk, so it's kind of just an overall general overview of what the field I kind of want to go into for my thesis work. So I'm going to be talking about graphical modeling and spatial interactions on kind of a subcellular level. So space, right? Oh, is this always cut off? I guess all my headings are going to be slightly cut off. So here's an overview, talk about kind of the goal of the research, or the first problem that we'll look to tackle, some of the data that is coming available here at OHSU, which is pretty exciting. A lot of this stuff is really cutting edge, including fluorescence microscopy, electron microscopy, super resolution fluorescence microscopy, and combining those two. And then I'll talk about some of the possible ways to model those things using different spatial statistics and graphical models, difficulties, and then thanks, and at the bottom this says pizza. All right, so motivation for doing this is sometimes cells go crazy and try to take over the world. One of the ways that they do this is through this EGFR signaling pathway. This is really, is there any way to change that screen? I'm going to miss all my sources and stuff.
You may want to try maybe check resolution.
If I just do...
Try 1024 by 768. Okay, apply.
Yeah, minimize that. Guess I should have checked this before I started, but it didn't occur to me.
I think it's because it's extended, but then you can't see your notes. I don't think they're...
Okay, I guess that's... All right. I guess there's nothing we can do. So EGFR signaling is kind of the big problem that the Joe Gregg lab has been working at. So all this imaging type data is kind of centralized around this idea. So this is a pathway that basically is how the cell, one of the ways that the cell decides to start dividing. So these receptors on the surface of the cell, EGFR receptors, they are bound by some growth hormone and then create this through a chain of interactions with other proteins, phosphorylations and dimerizations, have this signaling cascade going down into the nucleus, which initiates the proliferation of the cell. And this is a big problem because these receptors are mutated in a large amount of cancers, and normally mutated so that they're kind of stuck in the on position, which gives you the proliferative growth in cancer when you didn't want it. So it's mutated something in the signaling pathway. The HER2 signaling molecules up here, or some of these in that family, are mutated in about 20% of breast cancers. And not only are they mutated, but they become resistant to treatment in 90% of cases, and the regression-free survival time is only about 10 months. So this is basically like what we know about this signaling cascade at this point, where HER2 kind of dimerizes with HER3 or EGFR and has all these interactions with these different possible signaling molecules going down into the nucleus to turn on proliferation. We have drugs that target that stop the signaling, stop the binding of the molecule that induces signaling, but as I mentioned, like the cancers become resistant to this quickly. So clearly this type of picture isn't good enough to really, addressing this by itself isn't good enough to stop these cancers. So what's missing from this picture? Well, we just have like, okay, some stuff on the membrane, and then cytosol, this giant component of the cell that doesn't have any information on where exactly these different proteins are located. We don't know anything about organelles or vesicles or how these things are transported in there, what actually physically is going on. We can't see that stuff yet. Or we couldn't. We're starting to be able to now. So we need some data to try to understand these physical relationships. That's the wrong kind of data. So for starters, I guess, simplest approach is fluorescence microscopy. This is an image from the Human Protein Atlas where there's been a stain for the nucleus in blue, red is microtubules, and then the green is a stain for HER2. So you can see that it's localized mostly on the membrane, but it depends on the cell type, and the Human Protein Atlas has thousands of different proteins characterized in this way. But we still can't see organelles. We can see kind of generally where they're located in these two-dimensional pictures and kind of correlate the locations of different proteins, but we can't see individual interactions. We can't see dimerization. We can't see who's phosphorylating who or what complexes are built there and how they interact with the different compartments of the cell. So how could we see that stuff? Maybe with an electron microscope. This was the first one in North America. That's in Canada. The electron microscope can see on a much smaller scale because the wavelength of light is too big to see these individual particles interact. But the wavelength of electrons, if you think of them as a wave, is much smaller. So in an electron microscope, you accelerate some electrons, and instead of using glass to focus them, you use magnetic fields, and you pass those through a sample, and collect the electrons that either go through the sample or bounce off of it with different detectors. And then you can see at much smaller scales than you could see with light. So here's an image from an electron microscope looking at the same problem with the HER2. Another name for that is the ERBB2. And in this case, those particular proteins have been stained. Well, there's an antibody that recognizes HER2, and conjugated to that antibody is a gold particle. In this case, they're 10 nanometers, these little black dots. Each one of those is a tiny particle of gold. So we can see individual molecules because they've been recognized by this antibody with the gold particle. And this diagram here is like of one cell where on one side of the cell, this is pretty common in these types of images when you're looking at cells that have HER2 signaling. Part of the cell will have this kind of ruffled appearance with these filopodia-like projections. And maybe the other side of the cell will be flat. And this study found that the filopodia-like projections tend to have a lot higher concentration of these HER2 molecules. So there must be some connection between this ruffling and the signaling of the HER2 molecules. But we haven't really modeled yet the actual shape of the surface or how these connections are interdependent on each other. And the other thing with the electron microscope, right, it has to be in black and white. There's not different colors of electrons. So we can't do the same kind of staining thing to see different components of the cells. And there's only so many. I've only seen studies with maybe 2 different types of gold particles labeled of different sizes. So you can see maybe 2 individual proteins at a time, different classes of proteins at a time to see whether they interact with each other. But we can't go beyond that at this point. Some more on the microscopes. This is oh, and that was a 2D image, right? This is a Helios Nanolab machine that we have here at OHSU. And it's a focused ion beam combined with a scanning electron microscope. Sorry, yeah, combined with a scanning electron microscope. They call it slice and view. And this can see in 3 dimensions by taking your sample and kind of carve a trench out around the place that you're interested in. And then the focused ion beam can shave off incredibly thin slices of the sample, and then you image with the electron beam. So you take a picture, shave off a slice, take another picture, shave off another slice. And by doing that, you can build these 3D reconstructions of where exactly all these organelles and subcellular compartments are. So when you do that, this is the kind of image that you get off of that. Each one of these is a slice. As you can see, they're not all perfectly aligned, and they tend to have differing contrast levels. And we're actually moving through the sample. It's not refreshing as fast as it should, but we're moving through the sample with each new picture, which is hard to tell because they're that thin that the stuff doesn't even seem to change from picture to picture. The slices can be as small as 4 nanometers, which is on the scale of the size of a protein or a protein complex. And each pixel here is about 4 nanometers as well, so you get spatial resolution in all 3 dimensions that's about the same size, which is incredible. This is a reconstruction done by Danielle Jorgens and Jessica Riester, who obtained the images of breast cancer cell line. The one we saw previously was a pancreatic tumor. But this is a breast cancer cell line that's been grown in Matrigel, which is a 3D gel for mice that you can grow cells in to recapitulate kind of their full 3D shape. She was looking at these same Filopodia-like projections that we're interested in. And she goes through each one of these images and segments out using something like Photoshop to draw the outlines, and then can build 3D models of exactly what these shapes look like. So those protrusions are the Filopodia-like projections. The orange here is just degraded matrigel, and the yellow spheres are vesicles that are probably transporting some molecule in or out of the cell. We don't have any tag molecules here, so we don't know exactly what molecule we're looking at being transported in and out of the cell, but this is just to demonstrate some of the 3D capabilities. I can't even pause this because I guess I can. So I just want to orient you a bit too. Like, this is one nucleus here, and this is the border in between some cells. So one of the difficulties in here, I mean, she just segmented a small part of this here, but one of the difficulties is kind of telling where one cell ends and the other begins. So if we really want to characterize the interaction between these cells, a perfect segmentation of those details would be great. And there's people working on that, but it's a really tough problem. And I think that some of the segmentation can be combined with some of the modeling. We don't need to have absolutely perfect segmentation in order to be able to learn stuff from this. But this is a fast-growing area on how exactly to work with these images and characterize all the tiny things that is going on here. All right. Another electron microscope that we have here at OHSU from FEI. This one's a tomography microscope, and what that means is you take your sample and tilt it 70 degrees in either direction while taking images from the electron microscope, which you can then back project in order to construct a thinner 3-dimensional volume of the cell. So this is a reconstruction after the back projection and reconstruction of the 3D image. We're slicing through here and then outlining the different vesicles. In this case, the light brown is the outer double membrane vesicles. The pinker, smaller vesicles where some viral replication is going on. Brown is endoplasmic reticulum, and blue is some intermediate filaments. And green, you can see a little bit, that's Golgi. Oh, by the way, I forgot to mention, please ask questions if you have any while I'm going. Makes me more comfortable. So this is a study using electron tomography along with some of this gold nanolabeling done by one of our collaborators, Jan Post, who is at Utrecht University in the Netherlands. And the big gold dots here are just fiducial markers, so you can tell where you are in each image, so that you can align these three-dimensional, these different projections in order to tell where everything is. And then the small gold nano-label dots here are labeling calveolin, which is a marker for these calveoli, kind of invaginations from the membrane-bound vesicles as they're going into the cell. And he's trying to understand how certain molecules pass through the cell by coming into these vesicles, and then those vesicles moving from one side of the cell to the other. But you can see, in particular, this protein tends to, once you do the segmentation and 3D reconstruction, you can see how this protein tends to form along the outside of these vesicles, but not along the membrane in general. But even in his talk here, which was 2 weeks ago or so, he mentioned how there was difficulties in quantifying exactly the distribution of these individual particles and trying to understand where they are and where they aren't, what we would expect to see if they were randomly distributed and so forth. All right, so the problems with electron microscopy, although we can see 3D and in super high resolution, we can only see in black and white, and we can only mark one protein or maybe two proteins at a time. So maybe we can combine light microscopy with electron microscopy so that we can see a bunch of different types of proteins at the same time in different colors, as long as we can overlay the fluorescent image with the electron image. So this CLEM stands for Correlated Light and Electron Microscopy, which is up here, you can't see. But this is kind of how it's been used so far. You take an image where a protein of interest has been either tagged with an antibody that's conjugated to a fluorophore, in this case, green fluorescent protein, or some transgenic gene that expresses the protein that glows. And then you use that image to guide you in looking at the sample with the higher powered electron microscope, so that these are, areas of interest. In this case, this was viral replication again. So we can see the green gene was part of the virus replication mechanism, so we can tell that the virus is replicating in these areas, and then zoom in with the electron microscope to see, okay, is this happening in certain vesicles or in mitochondria or what? All right, but the resolution of light is not that great, right? That's why we're doing electron microscopy in the first place. But maybe we can find a way to go past that wavelength of light and actually use light to identify individual molecules. So that's the super resolution light microscopy. And the technique that's been used here by Shaolin Nan in Joe's group, who's heading this, is it's called PALM, for photoactivated localization microscopy. And the idea behind that is, like if we look at just these, say, fibers here at A, we can tell that there's two different fibers, but by B, the overlap of the spectrum from the two emitting fluorophores is so high that we can't really tell whether there's two things here or one, right? That's where our resolution breaks down. And so that's happening around the diffraction limit, which is about the size of a mitochondria shown here. And what we really want to see is individual proteins around the size of a couple nanometers. So how do we get this resolution to be higher? The idea is to get fluorophores that flash, basically, or can be excited by a certain wavelength in a controlled way, so that if we have two molecules next to each other that are both tagged with this protein, they won't always be flashing at the same time. So maybe we'll see a flash in one place and then another flash slightly next to it. And we collect a whole stack of these images and get a distribution of where these flashes are occurring. And assuming that that distribution is in a Gaussian curve, we can fit it to a nice smooth model and take the center of that curve to be the location of one in particular fluorophore. So we can separate two molecules that were so close together that we couldn't tell the difference from one image by using, through time, this flashing in a whole stack of images. And by doing that, we can take what would be, this is just one image from a fluorescent microscope, and get down to actually being able to see what a reproduction of, this is not just one image, it's a reconstruction of, from a whole stack of images of where individual molecules lie.
Nathan,
Yes?
can you clarify for them, stack of images from the same experiment or stack?
Same experiment, yeah. So it's the same sample, and while it's sitting there, the fluorophores are flashing, and you take a bunch of pictures. And each time it flashes, it looks like a slightly different position, but that distribution, the center of it, tells you where it's localized. Any other questions? Makes sense? All right. So this is the only study that I know about where it's combining electron microscopy with this palm method. And you can see in the background is kind of a low-resolution electron microscopy image. In the foreground, we have a higher resolution electron microscopy image. We're looking at one bacteria here. You can see its cell membrane. It's been segmented so that we can see these vesicles in white. In green are some filament bundles. In blue is this tubular structure that the researchers were interested in. And individual molecules labeled in the red and yellow from the palm technique can be localized exactly on that kind of blue structure. So it's this really combination of both of these techniques with super resolution light and electron microscopy, which is brand new. And the idea behind this is to be able to do this, we got, or Joe got a $1 million grant from the Keck Foundation, which is kind of the outline of what got me started thinking about this stuff. Okay, but part of that was how do we really understand these relationships between all these things, so how are we going to do the modeling? All right, so before we can really start modeling, we need to understand whether the spatial distributions make sense, or if they're if the particles that we're looking at are randomly distributed or not. So this is, on the left we have what's considered a complete spatial randomness. So this means that the probability of any two particles being within a certain distance has a Poissonian distribution. So this is the kind of background model of just saying if we were to randomly place dots on a grid, this was what, something of what they'd look like. This one looks more clustered, and but this one looks still pretty random, but it's not. So I'll explain that in a minute. So one way to measure that is by using Hopkins statistics or Ripley's K. And this is the difference between the expected value and the actual observed value of these distances. So in this, it's close to 0. We start getting some edge effects as we look at different larger distances, which is something that you have to take into account with these types of studies. But mainly we'd say this is randomly distributed because it's around 0. These guys look clustered because the distances, we see more particles at a smaller distance than we would expect by chance. And here we see less particles at given distances than we'd expect by chance. So the value is actually below 0, which means this is more, the particles are more regularly placed than we would expect. That's called a hardcore model. Because it could mean that there's something actually preventing these molecules from getting too close to each other, some type of hard core that we're labeling here. So these types of statistics can tell us about the distributions of the proteins, and then we can connect that with the subcellular features that we see from like the electron micrographs. And just an interesting side note is that a lot of these kind of things were developed in like forestry and geological techniques and trying to understand the three-dimensional distribution of, of different deposits of ore or gold, or in the forestry example, looking at where different types of trees are and whether they're clustered next to each other, which I thought was cool looking through the literature. So here's one study, again, looking at HER2 and whether it forms dimers. We're just looking at the surface of the cell here, and in different treatment conditions, we can quantify whether these HER2 molecules are more clustery than we'd expect to see by chance, and whether that kind of clustering changes with different treatments. So in this case, there's not much difference between whether the inhibitor is present or not. in the clustering of these HER2 molecules. In this case, the HER2 molecules have been double labeled with 2 different size gold beads. And this is their quantification of it. This is the actual distribution of distances between each other, and this is what you would expect by chance. So you can put some statistics on this. This is from the same study, but now looking at whether these HER2 molecules are forming dimers with HER3 molecules. So they've labeled HER2, it's best to just look at this picture, HER2 in the small nano gold dots, and the larger gold dots are HER3. So we can look at the distribution of both of these sets of dots and see if they overlap in a way. And, yeah.
On the question on your last slide, how many gold dots are represented in your distribution?
How many gold dots?
Yeah, where are you getting that distribution from?
Oh, so the distribution is the distances, pairwise distances between 2 dots.
How many?
All of them that you're looking at. So you look at all your dots, measure their distance to the closest one.
Well, the figure was like this.
Oh, so there's super small ones. Maybe you can't see them that well. So there's big ones and small ones. Okay. And this image isn't representative of all the data. This is just one image. All right, so we can look at the spatial distribution of these things and try to combine that with the different compartments of the cell or different subcellular features that we know are present based on the electron images, if we can put all this stuff together. So this is another spatial resolution. This is a paper by Shaolin Nan here at OHSU, looking at, in this case, RAS dimer formation. So this is without the signaling molecule, the RAS dimers or the RAS molecules are fairly evenly distributed. After the signaling molecule, they start to form these dimers, which can be quantified here using that Ripley's K measure, or he actually came up with another measure based on DB scan that was pretty cool. They can show that he's seeing an increase in the number of dimers more than you would expect by chance in these different conditions. But again, like in this, we're only seeing the super resolution microscopy images. These aren't overlaid with the EM images, so we don't know if these dimers tend to form preferentially in certain parts of the cell, whether they're on these Philopodia-like projections and things like that. Okay, so graphical modeling. One way to try to combine all these different types of information in order to make some coherent statements about what's influencing what is to use these kind of abstract graphical models. This is another image from the Human Protein Atlas, and it's just representative of some of the data that people have used for some of the spatial modeling so far. So this is from the Murphy group, which is out of Carnegie Mellon, and apparently he was formed the first computational biology program in the country. And it has probably hundreds of papers on trying to model these spatial interactions, but only from this fluorescence microscopy types of images. It's not super high resolution. So what they generally do is take their images and extract some type of features, sometimes on a per cell basis, sometimes on a pixel basis, of where things are located and what colors, what proteins are located next to each other. And the Human Protein Atlas has thousands of these, so you can look at whether certain proteins tend to be localized, their expression correlates between the two groups, and use that to get an idea of where proteins might be expressed in the cell. So you extract those features that might be about the size or morphology or intensity on this fairly broad level, and then build some model, whether it's a regression or a vector machine, hidden Markov model that you train with these features that you've extracted. And then that model, when you look at it and understand the dependencies in there, can tell you something about how these proteins are distributed, or classify proteins into certain types of distributions, or generate hypothesis on what interacts with what and what these things are actually doing. So this would be the same kind of general framework that I want to use, but the data, the images that I'll use will be different, and the types of features that I'll be able to extract also would be on a much higher, more granular level. One way of incorporating these spatial features is to do this kind of tiling. This is from a study looking at whole Drosophila embryos, where they did this triangulation of the space of the embryo and stretch things out a little bit so that they could get a feature vector basically of each of these triangles and where the proteins are located, and then correlate those spatial locations between different types of proteins. So I think something similar, although it'd have to be more flexible, would be possible looking at individual cells under different treatment groups, or looking at the expression of different proteins. So what is this graphical modeling stuff? So we have a bunch of quantities that we don't know that can have some value. So in this case, maybe this is a binary, whether the growth signal is present. And maybe this is some measure on how much the HER2 molecules are enriched on those Philopodia projections. And there's some complicated distribution, joint distribution on the values of all these different nodes. In this case, the gray nodes are things that have been observed, and the white nodes are things that we perhaps haven't observed, that we still might be able to infer something about based on their joint probabilities. So these are all connected in some way. If I change one of these, all these other guys change. This is a complicated thing to try to untangle, but given enough data to train the model, as you look through, you can perhaps whittle down these connections to find out which connections are really direct connections and which are indirect connections. So if there's not a line in between two things here, it means that they're conditionally independent. If I know what's going on with the other nodes that they're connected to, I don't need to know what's going on with the volume of the cell. For instance, in the Philopodia, all the information about the volume of the cell is being transmitted through this tortuosity or curvature of the membrane in order to change this value. So these can be trained with data, and in this case, this might give you some insight as to what's really going on here, whereas previously we might have thought, like, okay, the growth signal directly influences dimer formation, but maybe the model will tell us that, no, the growth signal only changes the ruffling of the membrane, and that has an effect on the amount of HER2, and that tells us how much these dimers will form. So we can use this for hypothesis generation and classification of different types of things and try to understand the full system in a way that might not be possible just by looking at it. This is an example of one of the earliest types of these graphical models from 1994 out of Stanford. So the previous one is kind of toy, and this is maybe more what they might look like with a huge amount of different nodes all interconnected. This is an overview of just some of these types of graphical models. So maybe you've seen naive Bayes before, or logistic regression. In all of these, the gray dots are things that are known, and the white dots are things that we're trying to understand or predict. So with naive Bayes, we have a bunch of knowns and one unknown that we're trying to predict. Same with logistic regression. But in the top row here, we have this directional where we believe at least that one node directly causes the other node to have a certain value. In the bottom row, we don't have that directionality. We just believe in some dependency between the values of each of the nodes. And each of these little squares, black squares, represents that there's just some function of the things that it's connected to that we incorporate in the model and are able to train and learn from data. In a hidden Markov model, we have more than one. We have these hidden set of states that we don't know the values for, and observations that we can see, and we're trying to infer the most probable set of states, ordering of states from those that we observe. Kind of similarly with a linear chain conditional random field, however, that can have dependencies on previous states, where the hidden Markov model, the probability of transitioning from one hidden state to another is fixed regardless of the observations, or regardless of where you are in the chain. We can get more general by allowing connections between our observed states or arrows in a nonlinear fashion between the unobserved states. and even more general by allowing higher order interactions more than just binary or directed interactions. In this case, we have 3 nodes influencing this one function, which is part of the decomposition of that probability, that joint probability over all nodes. So there's a bunch of different types of models, graphical models like this. People have been inventing new ones, it seems like, every couple weeks. But they all have the same idea of kind of decomposing this distribution across all the nodes and trying to understand the connections better. And they allow for more flexibility with more complicated models, and some of them even allowing you to add unobserved nodes as needed, which the more flexible the model, basically the more data that you need to train it normally. So which model I'll use to train this kind of depends on what the data looks like in terms of distributions of different proteins and organelles and such. Then I just have a list of some of these hidden logistic regressions, Bayesian nets, Markov random fields, discriminative random fields, hidden conditional random fields, and infinite conditional random fields, all of which, have the same general structure but get more complicated. So this is an example from some imaging, trying to understand image analysis using Markov random fields or discriminative random fields. And the difference between the two is a discriminative random field can take into account the neighbors of a specific node. So in the first case on the left here, we're trying to reconstruct, these are our original images. We add a bunch of noise, and then we're trying to get back the original image after, just by looking at the image with the noise. This is the result from the Markov random field, which just tells each pixel whether it should be black or white. And it makes that decision based only on what's inside the pixel itself, or small area itself. Whereas the discriminative random field can look at its neighbors and tell what's going on. So in this case, the job was to try to discriminate man-made objects from natural objects. And the discriminative random field allowing that, being able to look at its neighbors gives it more power in order to pull out these types of interactions. So a spatial model in cells would also have more power, being able to look at its neighbors and tell you, whatever outcome we're trying to look for. This one, is it? Yeah, it's a binary decision for this. Yeah, for this one.
So if you have
for these squares.
Heterogeneous field, which you have both, it'll still be forced into making it.
Yeah, it has to make a decision. Yeah, so it's like a logistic regression, but more complicated. All right, so once we have this kind of model where we can understand things, it might not be enough to just look at the model in order to say what's going on. So maybe we could do some type of simulation to see where we would expect, given this model, where we would expect to see certain proteins in some cellular model. This is from an older paper from 2008, where the kind of shape of the cell is set, but you can use partial differential equations to kind of explore where we would expect to see a distribution of different proteins. Some more advanced modeling that allows for the shape of the cell itself to change was done in this paper just in 2012. And they built these kind of three-dimensional models of cells that were where the shape of the cell depended on what was in each of the three-dimensional voxels. And these are fairly easy to access, so given some model, we'd be able to kind of simulate some of these things through some web interfaces. All right, that says potential difficulties. So the registration, which is just lining up the images, can be difficult, and the segmentation is a big problem. It's being worked on here by Guillaume, but I think that we'll be able to combine some of the modeling with the segmentation steps so that we don't have to have perfect segmentation, like I mentioned earlier. There's always, in building a model, you always have to kind of decide whether you want a discrete versus some continuous type of data. Most of these graphical models depend on some discretization of the data going into each of these nodes. So that will be a decision I'll have to make in building the model. And then training is always a big problem. You need some defined end goal that makes sense, and we need enough data to be able to train however complicated of a model I decide to use. So simplifying assumptions might be necessary. I might be able to use feature selection procedures or information from previous studies that we could leverage, or information from lower resolution images like Human Protein Atlas that we could leverage in order to understand some of these reactions or narrow down some of the possible graphical models as we're trying to train. And then interpreting can be difficult, but running simulations is one possibility to help interpret the model that I come up with. So overall, we're trying to move from this kind of idea of these signaling pathways to incorporate some real spatial aspects using these imaging data and this higher resolution imaging data that can be marked perhaps with a super resolution light microscopy of different species of proteins, so we can actually tell where things are, what's interacting with what, in what component of the cell, and really improve this model a lot in the hopes that we can start shrinking tumors and keeping them away for good and killing these rogue Napoleon cells and not letting them come back. So with that, I'd like to thank all my committee members and the Carbone lab and the DMICE department, my fellows for helping me listen to this talk and giving me advice, and Joe Gray for starting the impetus for this, all this really incredible technology that I'm really excited to work with. The imaging from Danielle that I showed you in the movies, and Damir and Michelle were also helpful to talk to about what type of projects are possible. My funding is from NLM. The Keck Foundation gave the grant that really spurred a lot of this work on the HER2 stuff. Imaging technology is all from FEI, and Intel is in the process of building a big cluster for us so that I'll be able to use that to actually train my models on these huge data sets. So any questions? And thank you. Kind of went fast. Yes.
So what are some of the biological discoveries and ultimately medical treatments that would emanate from, I would understand the value of understanding things, but what would potentially come forth from this?
So I think that I mean, in terms of like targeting certain therapies, so right now, for instance, for the HER2, like we have therapies that'll stop the HER2 signaling or stop it from forming these dimers, but that's not enough. Like the cells are finding a ways around that. So there's something that we're not taking into account, which is probably these spatial aspects. So there might be something that doesn't pop up in the GWAS or something that's mutated because it's a normal function of the cell that's being co-opted by some of these. So we have a different, maybe targeting it to a specific cellular component, or it may be understanding more about like how the cell morphology changes. That if we could, like Joe gives this example, like if we could cut off the filopodia, he talks about it like a power line. Instead of just trying to stop the electricity going through the power line, cut off the whole tower, and then you stop the signal transmitting. So I think that it just really opens up our eyes in terms of what to attack with a therapeutic, instead of just try to stop this gene from expressing or try to stop. We don't know where these dimers are forming. We don't know what's phosphorylating what necessarily or where all the time. So I think there's just a huge amount of possibilities in terms of targeting for therapies. Yes, Jess. Yeah, so it could be that it happens in different compartments of the cell, and perhaps, yeah. But I mean, I think like in order for any reaction to happen, like you need a certain concentration of those molecules to be present, right? So there can be, something about the cell morphology that allows for a higher concentration in a certain area that we haven't been able to capture before. We could think of that as an organelle, or it could just be like some, something to do with the morphology of the cell itself.
Thanks,
Yeah, I mean, you're right that it's not very totally formulated at this point. And I mean, I'm not going to be able to drive the acquisition of the imaging stuff. So it's more a matter of like what will be available. So in the Keck grant, I mean, they talk about the integrating the super resolution light microscopy and the EM images sort of around this HER2 signaling pathway. And then the piece of that that's kind of, at least in my opinion, very vague is how they're going to do the modeling reconstruction of that. And that's where I want to come into it. Like, how are we going to understand the relationship between those, you know, say we can tag 8 or 10 or 20 different proteins and understand their cellular localization in this pathway, in this HER2 pathway. Then we'll be able to see like when they interact with each other and what components of the cell, and how that differs between cancer and normal cells, or between the side of the cell that has the ruffling or the side of the cell that's flat. So I'm not sure if that totally answers your question, but I'm also not super clear on exactly what I'll be able to do given the data that's available either. Yes, Shannon.
So, single cell studies are kind of all the rage and are also under a lot of think about like single cell a lot of issues around reproducibility or generalizability.
Mm-hmm.
And so you're really focusing to some degree, how many, was it images from different experiments or multiple images from the same?
Sure.
You're very dense in terms of the image processing that you're doing in a single experiment. There is that question about reproducibility and generalizability. I was just wondering if you could talk a little bit about that and some things potentially.
Yeah, so I think that that's a really good point, and it's something that we definitely need to keep in mind. And I think one of the ways of doing that is to incorporate into those graphical models what we know about condition and such. So ideally, you know, we have different conditions, different cell lines. I mean, we're seeing differences just from the fluorescence microscopy between cell lines and localization of HER2 or whatever. So yeah, I mean, I think it's important to keep in mind that we need to look at a broad spectrum of different cell, whether it be cell lines or tissues, and to incorporate that in the graphical model as a predictor of the different behaviors that we're looking at. Yes, Ryan.
I was just going to ask how you see this, because you said that it'll help you do your training on the model. Is that because it's computationally intensive?
Yeah. Yeah, so I mean, also, I mean, dealing with these images, these are really high, like, you know, images are really high resolution, and if you want to get like a whole cell, you're tiling, you know, dozens of these images across each other, stacking up the three-dimensionality of it. It's just a huge amount of data to sort through. So extracting the features from the imaging modalities as a first step takes a lot of computing, which can be done in parallel, and is perfect for the type of cluster that we're building from Intel. And then secondly, training the model, incorporating information from all these, hopefully a lot of different imaging sources and different conditions and different signaling.
So is a lot of that stuff already in place, or are you going to have to
Yeah, there's some stuff in place, and yeah, there's people working on it in terms of the image segmentation and stuff. There's people here that are working on parallelizing. I mean, for the most part, there is some parallelized code, but it's pretty young. Like it's not fully mature. But yeah, that's a concern. I mean, I guess in general, I'm a little worried about like, you know, waiting for certain steps in order to go next to the modeling. But I also think that it's worthwhile to kind of simulate some type of data in order to build models that make sense first, or use lower resolution data from Protein Atlas or from some of the microenvironment arrays or something like that to build similar models before, if it's, you know, we're really finding that this is taking a long time to get the type of data that I'm hoping for. Yeah.
It's just kind of interesting because image data that we're trying to hook up to.
Yeah.
So a lot of what you're talking about here, I could see applying.
Yeah, for sure. Yeah. Yes, come on.
So you remarked that you won't have a lot of control over like what experiments are going to be done. Like say if they're going to be after certain cell lines or drug combinations. But don't you think that once the initial sort of thing comes out and somebody will need to generate hypotheses, right? So like your models could actually, if they sort of integrate the data and suggest new things, don't you think then those could lead to new generation, new hypotheses and new experiments?
Yeah, absolutely, and that's one of the advantages of the graphical model. I mean, understanding that can really lead to hypothesis generation, and yeah, I mean that would be ideal.